pancreas. Examination questions. Location of liver and pancreas. These glands release their secret through the main ducts into the duodenum. Liver located right side in abdomen cavity. Pancreas could not be visible because it's located under the stomach. Liver carry out many functions: synthetic, synthesis of glycogen, proteins for blood, bile, protecting and antitoxic, store of lipid-soluble vitamins, hematopoiesis in embryogenesis. Liver, the biggest gland in the body. Its weight about 1.5 kilograms. Its parenchimal organ consists of stroma and parenchyma. Stroma represented by thick connective tissue capsule and loose connective tissue septa, which divide an organ for lobules. Liver lobules is a functional structural unit of the liver. It 1-2 millimeters in size, 6-angle pyramid in shape, and there are about 500,000 of lobules like that in the liver. So in the section it looks like spongy. To understand the structure of liver, it's necessary to remind the blood circulation. There are 3 parts in it. Inflow of blood into the liver take place from 2 sources: the portal vein, which collect the blood from all unpaired organ of the abdomen cavity, including stomach and intestine, where absorption of the digestion of food products take place. In the stomach and intestine absorbed the drugs and poisons if enter this food. And these bloods go through the liver. The second source is hepatic artery, which starts from the abdomen aorta and bring the blood enriched with oxygen. These 2 vessels go together and branching together, initially for lobular vessels, then for segmentous vessels, and then to interlobular around the hepatic lobule. Interlobular vein, artery, give the small vessels around lobule, and artery give the arteriole and vein, small venule. They fuse together and compose the sinusoid blood capillaries inside the lobule, which bring the mixed blood, which flow from the periphery to the center of lobule. It blood circulation inside lobules. Then blood draining include central vein, sublobular veins, then collecting veins, and then 3-4 hepatic veins which enter the vena cava inferior. This picture demonstrates the liver lobule of pig. There is a central vein in the center and well-developed interlobular connective tissue. So it's quite easy to find the lobules, hepatic lobules, under microscope. In human liver there is central vein but not good developed connective tissue around, so it's much more difficult to recognize lobules in the human liver. This is a figure of human liver, connective tissue capsule around and lobules. In the center of hepatic lobule there is central vein and radially going sinusoid blood capillaries around lobule, interlobular artery, vein, and bile duct. These three vessels combined into hepatic triad. 3 vessels, triad, and there are up to 6 triads around every lobule. So this is one hepatic lobule, this is another one, the half of next is a piece of 2 others. This picture demonstrates the one hepatic lobule, the central vein and sinusoid blood capillaries is mixed blood which flow from periphery to the center. It's derived from interlobular artery and vein. Between sinusoids located the hepatic cords, which consists of 2 lines of hepatocytes with bile canaliculi inside. In the bile canaliculi, the bile flow in opposite direction from the center to the periphery of liver lobule and enter the interlobular bile duct. This microphoto demonstrates the histological preparation of liver lobule after injection of liver blood vessels with blue dye. Central vein and sinusoid blood capillaries filled in by blue dye. High magnification. This is transverse section of hepatic cord. It consists of 2 hepatocytes with nuclei and bile canaliculi between. So these poles or surface which secrete the bile called the bile surface or canalicular surface or poles. Hepatic cord surrounded by sinusoid blood capillaries, both sides. This is a blood flow inside sinusoids. It's all linked by fenestrated endothelial cells which contain no basal membrane. Between the sinusoid and hepatic cords there is a cleft-like perisinusoidal space of Disse where the blood plasma circulates and components of blood can enter this space for absorption into hepatocytes, and in this region the blood proteins produced and they release into the space of this. So in these regions the metabolism, exchange of substances between blood and hepatocytes take place. To facilitate this exchange of substances, in these poles hepatocytes have microvilli. These poles connected with sinusoid blood capillaries called the vascular poles. In the sinusoid wall there are also Kupffer cells or stellate macrophages which can phagocyte bacteria and foreign particles which flow through the sinusoids. In these vascular poles the glycogen trophic inclusion produced and accumulated. Glycogen synthesized from glucose which enter the space of this from blood which flow through the blood capillaries. In smooth endoplasmic reticulum here synthesized glycogen, and when necessary it catabolized, destroyed to glucose, and glucose can enter the blood circulation. There is also another type of cells, ethos cells or perisinusoidal lipocytes. These cells contain the droplets of fat where the lipid soluble vitamins can accumulate it. Vitamins like vitamins A, K, D, E. So before the liver is a store of vitamins. This is a sinusoid blood capillary transverse section. Its wall consists of endothelial fenestrated endothelial cells and star-shaped macrophage Kupffer cells located here. There is the perisinusoidal space, microvilli or hepatocytes, between hepatocytes bile canaliculi, perisinusoidal lipocytes, ethos cells. In addition to accumulation of lipid-soluble vitamins in unfavorable conditions after the action of toxic substances, it can produce collagen, and in the space of this cell initially appeared the collagen fibers. It initiates the connective tissue growing and liver cirrhosis take place. The functions and blood circulation disturb, and finally the liver destroyed and the patient will diet. This picture demonstrates sinusoid blood capillary lining by endothelial cells, and there is star-shaped macrophage Kupffer cell, perisinusoidal lipocytes, and additional pit cell, big granular lymphocytes, natural killer, which can release cytokines which killed defected hepatocytes. At the same time, it produces stimulator of regeneration, proliferation of normal hepatocytes. It helps to the liver regeneration after disease or some disturbances. You can see Kupffer cells if you inject, for example, the black dye into the blood circulation. This dye go through the vessels and disappeared, but during it go through the sinusoid blood capillaries, Kupffer cells phagocyte the droplets of dye and accumulate it in cytoplasm, so make Kupffer cells visible. It was the classic lobule description, classic point of view. The liver consists of 6 angles pyramids with central veins in the center of classic lobule. Another point of view, that liver consists of portal lobules, 3 angles connected the central veins of classic lobules. They straight with interlobular duct in the middle. Liver acini, oval in shape, include 2 central veins and branches of 1 hepatic artery which supply this triangle region of 2 neighboring classic lobules. The closer to the center of acinus, the better supply of oxygen. This region first affected by poisons coming here from the blood of portal vein. This is a bile canaliculi between 2 neighboring hepatocytes in liver trabecle. So bile canaliculi has no its own wall. It composed by the grooves of 2 neighboring hepatocytes which connected strongly these desmosomes, and you see also the microvilli which increase the exchange of substances, the square for diffusion of bile components from biliary poles of hepatocytes. So bile never reach the blood and sinusoid blood capillaries located around hepatic trabecles. But if hepatocytes are destroyed, the bile can reach. The preparation and histochemical preparation stained for alkaline phosphatase determination. You can see the bile canaliculi inside liver trabecles. It's because the biliary poles of hepatocytes possess the high activity of this enzyme. Bile ducts start from interlobular ducts, then fuse to segmental ducts, left and right common hepatic ducts, which continue to main duct, which release bile into the duodenum, or the bile can flow partly through the cystic duct and accumulate it in gallbladder. Gallbladder is tubular organ. Its thin wall consists of three membranes: mucous membrane, muscle, and serous membrane. Mucous membrane composes numerous folds visible without microscopes. Every fold contains the connective tissue core covered by simple columnar epithelium. It contains microvilli and can absorb the water from bile, and it became more concentrated. Is the reason why here bile stones consist mostly from cholesterol. So I describe you shortly the structure of liver from organic through the tissue and cellular to subcellular levels. Pancreas is parenchimal organ. It consists of tail, body and head, which located inside the duodenum semicircle. And this is common pancreatic duct which releases the secret into duodenum together this bile duct. So it's parenchymal organ which consists of stroma and parenchyma. Stroma represented by very thin lucid capsule and connective tissue septa which divide an organ for lobules. They are visible here. It's a gland of mixed secretion. It consists of exocrine and endocrine parts. Exocrine part composed 97% of parenchyma. It is the compound tubular alveolar glands which consist of secretory portions which produce the secret and ducts which release the secret. Every acinus is a vesicle which lining by acinocytes, secretory cells. They are pyramidal in shape, two zones, the basal, basophilic or homogeneous zone under the nuclei or between the nuclei. It's basophil because it contains rough endoplasmic reticulum, because main function of these acinocytes production of proteins for export. It produces precursors of four main digestive enzymes: trypsin to digest proteins for amino acids, lipase which digest lipids for glycerol and fatty acids, and it digests carbohydrates to monosugars by amylase. The precursors of this enzyme accumulated in Golgi and in granulars in apical portion of cells, and they release into the lumen of acini when it necessary to digestion of food. And apical portions contain Zimatkin granulars. its oxyphilic zones. Zimatkin granulus with precursors of enzymes. The system of ducts start with intercalated duct. Sometimes it enters the acinus, so-called centroacinous cells appeared, small cuboidal or flat cells. It continued to intralobular duct, which collect the secret from all acini of the lobule. Then interlobular duct, which located in interlobular connective tissue and collect the secret from several lobules, and then main duct, which release the pancreatic juice into the duodenum, and there it participates in the digestion of food. Endocrine part of the pancreas compose only 3% of the parenchymal volume. It consists of pancreatic islets of Langerhans glands, and there are 1-2 millions of them. They scattered among the acini. Their size between 0.1 and 0.3 millimeters in size. Their cells much lighter and divided into 5 types of endocrine cells, which produce 5 main hormones. This is the islet of Langerhans glands. The most numerous are beta or B cells from basophilic. They compose up to 70% of insulocytes and produce hormone insulin, which stimulate the absorption, the uptake of glucose from blood by tissues. If no insulin, tissues cannot uptake glucose for the metabolism, and the level of glucose in blood increase. The excess of it release this urine, so it takes place in disease called sugar diabetes. Alpha or A cells from acidophilic compose 20% of cells of islets. They are located at the periphery and produce glucagon. Glucagon acts in opposite direction to insulin. It increases the level of glucose in blood because it stimulates formation of glucose from different sources and stimulates destruction of glycogen to glucose. D cells from dendritic cells contain processes. There are up to 10% of them in the islets. They secrete somatostatin. D1 cells, these single, not numerous endocrine cells, produce vasointestinal polypeptide. And PP cells, this yellow, produce pancreatic polypeptide. These 3 hormones regulate the activity of exocrine part of the pancreas as well as other neighboring organs of digestion.